Hi, my name is Zoya Cerrone, and my research explored the relationship between psychosocial adversity and executive functioning through fMRI. I researched in the Gab Lab under the advising of Dr. Nadine Gab and Dr. Theodore Tureski. So, why is the relationship between psychosocial adversity, or PA, and executive functioning, or EF, an important one to consider? PA, which includes abuse, neglect, and household dysfunction, is a severe stressor, and over 1 billion children in the world have experienced some form of violence, which is a single form of PA. Exposure to PA can produce a variety of physiological and behavioral changes, which can lead to alterations in brain structure, function, and connectivity. Exposure to PA is associated with many adverse cognitive consequences, including impaired EF. EF is a set of higher order cognitive processes that serve as a predictive measure of academic and professional outcomes in adulthood. One specific type of EF, response inhibition, focuses on behavioral regulation and is widely researched as a measure of EF. In summary, PA is associated with impaired EF, which can result in adverse long-term consequences, especially when experienced in childhood. Though this relationship has been explored in the literature, most studies have explored this relationship behaviorally, and those that have used neuroimaging have typically had very small sample sizes and have worked with adults, even though EF rapidly develops between the ages of 3 to 8. Additionally, most of these studies have been conducted in high-resource environments, even though low-resource environments are found to have high levels of PA. My project aims to fill these gaps by exploring the relationship between PA and brain substrates of EF in 71 children 5 to 7 years of age via fMRI data collected during an EF task in an environment of extreme poverty in Dhaka, Bangladesh. Though our group is not well represented in current literature, we hypothesized that since we were using a widely validated EF task, we would observe activation patterns consistent with literature, for example, in the prefrontal cortex, the anterior cingulate, the thalamus, and the insula. And there would also be large clusters of these activated areas and a lack of refined networks as we were working with young children. In determining how PA would alter the activation in the EF task, we hypothesized that we would have decreased activation in key response inhibition regions, as this is what previous literature has found, though they were using a different population than ours. We also hypothesized that there may be some sex effects, but age would likely not explain the variance in activation. After pre-processing and processing our data, we found that the activation patterns for our group for the EF task resembled current literature, where we had activation in the prefrontal cortex, the supplementary motor area, the insula, as well as the fusiform and occipital areas. For the adversity analysis, we found significant clusters for the abuse, household dysfunction, and total psychosocial adversity, which was a composite measure of the other three in these analyses. For the abuse and household dysfunction measures, we found higher activation in individuals with exposure to PA in the anterior cingulate cortex, which is important for behavioral regulation. While for the total PA analyses, we found higher activation in the lentiform nucleus, which is part of the basal ganglia that works to inhibit motor movements from the cerebellum. You can see these activation areas a bit clearer in the video version. It's important to note that while previous literature that worked with adults or in individuals in higher resource environments typically found decreased activation in EF-related regions when considering exposure to adversity, we found higher activation in these areas, which could be a sign of overcompensation, where overcompensation occurs when there is impaired cognitive processing. Additionally, using sex and age as covariates of no interest to see if sex or age may explain the variance we found from the adversity analyses, we found that sex and age did not affect the significant results, giving us more confidence that the results we found in brain activation alterations were due to our adversity measures. As this is the first project to examine how PA is related to brain and behavior associated with EF in young children in a low-resource environment, there are many implications for these results. Not only does this allow us to visualize the alterations that occur in brain activation when looking at previous exposure to adversity, but also highlights the necessity for more research in this low-resource environment being done, so that these results can be compared with future results, ideally in similar resource environments or age groups, to gain a more comprehensive understanding of what may be responsible for these differing patterns of activation. Additionally, these activation patterns highlight the importance of conducting longitudinal studies and intervention-based studies to understand how these networks develop and are activated over time, as well as if any changes that are visible from adversity are reversible through intervention. Thank you.